All right, let's talk about turmeric. You know, that vibrant yellow spice we've been told for ages is a health superstar? It's loaded with this stuff called curcumin, which is famous for being anti-inflammatory, antimicrobial, you name it. But it turns out a recent scientific discovery has thrown a massive twist into that story.
What if that hero compound we've all been celebrating, curcumin, is actually more like a disguise? You know, like a superhero's secret identity? What if its real power only gets unleashed after it changes into something completely different? That is the amazing puzzle we are going to unpack today.
You know, every great story needs a villain and a hero. In this story, the villain is a really nasty bug that can live in our stomachs. And the hero? Well, that's where things get really interesting. So, let's meet the player, shall we?
First up, our villain, Helicobacter pylori, or H. pylori for short. And this thing is everywhere. It infects more than half of all people on the entire planet. It sets up shop in your stomach lining, and for a lot of people, it's the main cause of painful ulcers and can even lead to stomach cancer. So yeah, it's a huge deal, and finding new ways to fight it is super important.
So how does H. pylori actually do all this damage? Well, it has this main weapon, a protein called the VACA toxin. The best way to think about it is like a microscopic hole-punching machine. Seriously, it grabs onto our stomach cells and just starts punching these tiny holes or vacuoles into them. And all that damage is what leads to the inflammation and disease. So if you want to stop the villain, you've got to shut down this weapon.
And that brings us right back to our supposed hero, curcumin. Scientists already knew it could fight H. pylori, but they noticed something kind of weird. The way it was working just didn't quite add up. And that led them down a rabbit hole to this fascinating puzzle that pretty much challenges everything we thought we knew.
Okay, so here's the mystery. When curcumin gets inside your body, it's not very stable. It breaks down or oxidizes really fast. And this is what sparked a brilliant idea for the researchers. They thought, wait a minute, what if curcumin itself isn't the active ingredient? What if the real heroes are the other compounds it turns into right after it breaks down?
So before they even touched a single test tube, the researchers went digital. They ran these incredibly powerful computer simulations, it's called in silico analysis, to see how well curcumin and all its different oxidized forms could physically grab onto and disable that nasty VACA toxin.
And whoa, check out what the computer spit out. This chart shows a binding score, which is basically just a measure of how tightly a molecule can glom onto the toxin. And a higher negative number here means a way better grip. As you can see, regular curcumin, yeah, it binds okay, but just look at its oxidized product. Its score is way higher. It latches on so much more effectively.
Let me put it this way. Think of that VACA toxin as a lock. The computer model showed that curcumin is kind of like a key that fits, but it's a little loose, a bit wobbly. It works, but it's not perfect. But that oxidized product? Oh, that's like a key that was custom made for the lock. It slides right in, fits perfectly, and completely jams the whole mechanism, shutting down the toxin for good.
Okay, the computer models looked amazing, but a simulation is still just a simulation. They needed real-world proof. So the researchers moved from the digital world to the lab bench, and they designed this really clever experiment to see if curcumin truly needs to transform to get the job done.
The setup for this experiment was just beautiful. They took human stomach cells, split them into groups. One group got normal curcumin, the kind that can break down and oxidize. A second group, and this is the key, got a chemically locked version of curcumin. It was tweaked so it could not oxidize. And a third group was the control, nothing added. Then they just unleashed H. pylori on all of them and watched to see what would happen. This was it, the moment of truth. Would that locked curcumin still work? Or was the transformation, the oxidation, the real secret ingredient? The results from the lab were about to give them the smoking gun.
And the answer wasn't just buried in some data sheet. Nope, you could actually see it with your own eyes, just by looking at the cells under a microscope. The evidence was absolutely unmistakable. And this table just lays it all out perfectly. The control cells, the ones with just H. pylori, totally wrecked. You can see the vacuolation, all those holes we talked about. Now get this, the cells with the locked curcumin that couldn't change, they were also heavily damaged. Nothing happened. The only group that was protected were the cells treated with normal curcumin, the one that was free to transform into its powerful oxidized form.
So the takeaway here is crystal clear. Curcumin itself isn't the direct hero of this story. It's more like the mild-mannered alter ego. It has to go through that transformation. It has to oxidize to become the superhero that can actually fight the toxin. It really is a classic case of a secret identity.
And hey, don't just take my word for it. The researchers themselves put it perfectly in their paper. They said that both the computer data and the lab data point to the same thing. It's the oxidative products that are doing the heavy lifting here and stopping that vaca toxin in its tracks.
This discovery is absolutely incredible. And it really makes you wonder, right? If curcumin has been hiding this secret all along, what other natural compounds are out there that might be doing the same thing? Things like the catechins in green tea, are they also working through these powerful, transformed versions of themselves? It just totally changes how we might go about looking for new medicines in nature.